Okay, we're going to have to start. So, fermentation pathways. Okay, you start with organic carbon, you don't have oxygen, generate ATP and ADH by fermentation. Okay. What if you do have oxygen? Then bacteria will perform respiration. So what is happening during respiration? Well, depending on the carbon source, but let's just stick with glucose. Okay. Then the first step is going to be glycolysis. You'll get pyruvate, just like we saw before. And then instead of using pyruvate as the electron acceptor for NADH to produce a fermentation product, pyruvate is going to be converted to acetyl coenzyme A. So one carbon dioxide is kicked out, so this is 2 carbons in a reduced form. It will be attached to oxaloacetate, 4 carbons, to give you citric acid, 6 carbons. And then, little by little, this citrate is going to be cycled around to oxaloacetate again. And once more, you're going to see the Krebs cycle in biochemistry later on this term. So for right now, you don't have to worry about the structures of the intermediates. But what happens through this cycle is that, okay, both of these carbons here get oxidized and expelled as carbon dioxide. Since they are oxidized, that means that the carbon here is losing electrons. And these electrons are transferred onto NADH and FADH. So you generate a lot of NADH. And then because you need to regenerate NAD plus for glycolysis to work, the electrons are going to be transferred from NADH to an electron transfer chain, which eventually puts them onto oxygen. So you have these 3 main steps: glycolysis, the Krebs tricarboxylic acid cycle, and then electron transport and oxidative phosphorylation. And it's the oxidative phosphorylation step which will generate a lot of ATP. Okay, so if you think about glycolysis and the Krebs cycle, then the bacterium will have generated a lot of NADH, and it's got to get rid of these electrons. So here, NADH is going to be, recycled by NADH dehydrogenase. So you get NAD+, and NADH dehydrogenase will transfer these electrons onto quinone, or coenzyme Q. So this is a lipid-soluble molecule which cycles in the bacterial plasma membrane. So Q here is the oxidized form, QH2 is the reduced form. So it's picked up the electrons from NADH, so it's reduced to QH2 at the cytoplasmic face of the membrane. And these electrons are then going to be transferred onto cytochromes on another protein complex. And when that happens, the protons from the QH2, the reduced form, are liberated on the periplasmic face of the membrane. So this is an important step because it generates a proton gradient. Because initially, the protons are here in the cytosol. They're transferred onto quinone along with the electrons at the cytosolic face. And then they're liberated at the periplasmic face. So the coenzyme Q, or the quinone cycle, is one of the parts of the electron transport chain that generates a proton gradient. Now the final step in this is transfer of the electrons through to different cytochromes, and finally cytochrome oxidase will transfer the electrons onto oxygen to generate water. So harmless waste product. In E. coli, there are two cytochrome oxidase complexes, one which is called BO down here, which is used when there is a lot of oxygen present, and it's used preferentially because the BO cytochrome oxidase can also transport protons across the membrane and contribute to the proton gradient. Now, if there's not a lot of oxygen around, then the BD branch will be used. So the BD cytochrome oxidase has a much higher affinity for oxygen. So it will function under conditions of low oxygen, but it does not transport protons across the membrane. So that's why these two systems exist in E. coli. Now, once the proton gradient has been formed, then it can be used to generate ATP by the proton-dependent ATPase, which is in the membrane. So protons that are in the periplasm transported across through the proton-dependent ATPase, down the gradient, and this is used to generate ATP. Now, the big advantage of respiration compared to fermentation is the ATP yield is much higher. So the maximum, if there's really a lot of oxygen present, is 38 ATP per molecule of glucose. So that's, almost 20 times more efficient in terms of ATP generation. So E. coli has got an electron transport chain like this in its membrane. Our mitochondria function in the same way. Many aerobic bacteria will be using this mechanism. So what about if you don't have oxygen present? Okay, fermentation is a possibility, but some bacteria can perform what is called anaerobic respiration. Anaerobic respiration. Think about that for a minute, because it sounds like it should be impossible, right? Respiration, you breathe, you use oxygen. But in this sense, it just means electron transport onto an inorganic electron acceptor. So nitrate can be used as an electron acceptor instead of oxygen during anaerobic respiration. So Parococcus denitrificans can do this. It can respire without oxygen. So what is happening here? This is the electron transport chain, for example, in Parococcus denitrificans. You start off with NADH, which transfers its electrons onto coenzyme Q, quinone, which will then transfer the electrons into another cytochrome big complex here, and finally, in the periplasm, and other cytochromes will transfer these electrons onto nitrate. It's going to be reduced to nitrogen gas, dinitrogen. So this is one pathway of anaerobic respiration using nitrate as the electron acceptor. Other bacteria can reduce nitrate all the way to ammonium. Okay. So if nitrate is reduced to ammonium, then it remains in a form that can be easily assimilated into by other bacteria into form amino acids. However, if this pathway is used, then dinitrogen, so nitrogen gas, is going to be released. This is not easily assimilated into organic molecules. So this is a pathway which is called dissimilatory nitrate reduction, because the nitrogen that is formed cannot be assimilated. So it's released into the atmosphere. Okay, so why is this possible? It's possible because nitrate to nitrite and nitrate to nitrogen gas have a positive redox potential as half reactions. And our electron donor, NADH up here, has a redox potential of minus 0.32 volts. So electron transfer from NADH to nitrate is highly favorable. So that's why this process works. Okay? The electrons are being transferred from a very good donor to a pretty good electron acceptor. Not quite as good as oxygen. Okay? And this is also true for other inorganic electron acceptors. So some bacteria can use nitrate as the electron acceptor. Others can use sulfate. Some can use, what did we find out? Did anybody find this out since last time? This is ferric iron or ferrous iron, iron 3. Oh man, we forgot since yesterday. Let's say this one is ferric, and that's ferrous. So iron 3 plus can be used as an electron acceptor. Arsenate, selenate, if they're present. So all of these inorganic compounds can be used as electron acceptors in the absence of oxygen to perform anaerobic respiration. So in underground aquifers, if there is organic carbon, the bacteria that are present will grow by anaerobic respiration. First, they'll use up all the oxygen, then all the nitrate, then all the manganese, then all the iron, then all the sulfate. So the best electron acceptors will be used up first. When I say best, that means that the half reaction is very, very highly positive. So you use up the oxygen first, then nitrate, then iron, then sulfate, then the other ones that are progressively less favorable electron acceptors. And the last of all is carbon dioxide, which is the worst electron acceptor, only used by methanogens, which are archaea bacteria. Okay, so for the lab classes, which are in a couple of months, you have to remember this one because anaerobic respiration using sulfate as the electron acceptor will produce hydrogen sulfide. This is going to be important in one of the biochemical tests that you're going to use in the lab classes. Okay, so those are the two types of metabolism that chemoheterotrophs will use. Fermentation pathways, respiration with either oxygen or some other inorganic electron acceptor. And the input, it all starts with organic carbon as the energy source and the source of electrons. Chemolithotrophs. So basically, the source of electrons is inorganic. This will be the base of their source of energy for chemolithotrophs. And they will all fix inorganic carbon. Three different types of chemolithotrophs: hydrogen oxidizers, nitrophile bacteria, and sulfur oxidizers. And in these two cases, nitrophile bacteria and sulfur oxidizers, they are performing the opposite reaction to what we just saw, anaerobic respiration. So they will use reduced forms of nitrogen or sulfur as the electron donor, and the electron acceptor is oxygen. So these only occur in the presence of oxygen. Okay, why? So here you've got nitrophile bacteria They might have nitrite as the electron donor. The electron acceptor is oxygen. This will work, okay, because you're going from, well, okay, nitrite is a pretty poor electron donor. It's not good. It's pretty good electron acceptor, but oxygen is even better. So the delta V here is going to be plus 0.4 volts, which will give you a negative delta G. It's favorable. So that's why this can work, okay? Previously, sulfate and nitrate were used as the electron acceptors, and that works because there's no oxygen present. If you have sulfide, nitrite, or ammonium, and oxygen, then you can generate energy by oxidizing these reduced sulfur and nitrogen compounds. So let's have a look at a couple of examples of these different types of bacteria. Hydrogen oxidizers. This works if you've got a source of molecular hydrogen. So in hot springs, sometimes, volcanic springs, hydrogen gas can be released from white smokers under the sea, or hot springs that are on the surface of the earth. You can have hydrogen gas, oxygen is present, produce water. So aquifex, making water. The people who invented the genus names here were like having a lot of fun. Hydrogenophaga eats hydrogen. So, and then methanogens are also an example of chemolithoautotrophs. Molecular hydrogen is the electron donor. Carbon dioxide this time is the electron acceptor. How does the energy metabolism work? So if you look at the redox potential of this half reaction, your E is minus 0.42 volts. So molecular hydrogen is a better electron donor even than NADH. So electrons can be transferred directly from molecular hydrogen to NAD+. And that will generate NADH directly. Then you can use the electron transport chain to put the electrons onto oxygen. And this will allow you to produce ATP. So you get all the three forms of metabolic energy that you need. Hydrogen onto NADH, electron transport to produce proton motive force, and then ATP. Okay, that's for these guys if you have molecular oxygen present. In methanogens, the whole thing is way too complicated and very different. Nitrifying bacteria. So there are two types, Nitrosomonas and Nitrobacter. So Nitrosomonas will take ammonium and oxidize it to nitrite. And Nitrobacter will take nitrite and oxidize it to nitrate. Remembering which is which, okay? Nitrosomonas, ammonium. Okay, so remember this one. Nitrosomonas takes ammonium. Nitrobacter is the other one. Oxidizes nitrite, bacter, nitrate. Now, in this case, how is energy metabolism going to work? Because with this half reaction, nitrate is a good electron acceptor. Nitrite is not a good electron donor. So you can't transfer electrons from nitrite onto NAD+, because you have a very, very unfavorable electron transfer here. Look at the scale up here. What you're saying is, we're going to transfer electrons from way down here all the way up to there. No, can't work. Only works this way. Okay, so this cannot happen directly. So how do these bacteria produce NADH? What can they produce? So these electrons can be transferred onto oxygen, right? What can the bacterium produce during that process? It's going to help it get the rest. Yeah, but that's the end product. But during the intermediates, okay, then you'll release heat energy. It'll be nice and warm, but they will not be able to perform any biochemistry. What's happening here, for example? Oh, look, here's a nitrate to nitrite step there. But we want to make this go the other way around. What could occur? You have electrons, they start off on the nitrite, okay? And they're going to be released, and they're going to be transferred actually onto where? Oxygen, right? Yeah, okay. During this transfer, what can the bacterium do that will allow it afterwards to generate ATP and NADH? Yes, yeah. So somehow during this electron transport chain, they've got to get protons across the membrane. So you start off with a redox reaction, which is really kind of pathetic in terms of, energy terms. But it's enough just to get a proton or two across the membrane. And if you can transfer enough of them, you can produce ATP. And if you've got a big proton gradient, remember way back to the Nernst equation. If your gradient is very high, that can give you a good electronic potential. So that's enough that you can get an electronic potential. There will be enough to drive this reaction. So first you've got to have transfer of electrons onto oxygen through an electron transport chain. This allows you to generate a proton gradient, and that will allow you to make ATP and then drive the production of the reduction of NAD plus to NADH by reverse electron transport. It's going the kind of wrong way round, or rather in the opposite direction compared to what we saw with respiration. Okay, sulfur oxidizers. This can occur aerobically or anaerobically. So anaerobically, thiobacillus denitrificans, the electron donor is sulfide or molecular sulfur. The electron acceptor is nitrate. Thiobacillus ferrooxidans, if you've got iron and sulfide, these can be oxidized to, ferric iron and sulfuric acid. And both of these processes are thermodynamically favorable. This one, thiobacillus ferrooxidans, is responsible for acid seepage from old iron mines. So this can be a serious problem. So it has been a serious problem in the States. And the bacteria in these old mines produced millions of tons of sulfuric acid that leached out of the old mines and polluted the water. And the solution to this problem was realizing that the bacteria require oxygen. So you just block the mine up, block the mine entrance, so they don't have access to oxygen anymore, and it solves the problem. One of the things here is millions of tons. Why is that the case? Because all of these metabolic reactions here, the delta G is really very, very small. So bacteria, these chemolithoautotrophs, are really scratching a living from not very much at all. So they need to oxidize a lot of iron and a lot of sulfide just to get a few molecules of ATP. So they really use up a lot of their inorganic substrates. And that's also true for the nitrogen oxidizers, especially nitrobacter. This is really kind of a pathetic chemical reaction to try and live on. Right? So they need to use a lot, and they grow slowly. Oh yeah, the other thing about sulfur oxidizers is that they have a kind of a cute way of producing ATP. So adenosine monophosphate can be sulfated to give you adenosine phosphosulfate, and then sulfate groups can be switched for phosphate groups, and this gives you ADP. And then you switch the phosphate groups around, that gives you one AMP and one ATP. So sulfur oxidizers can directly produce ATP. Okay, so all of this, like anaerobic respiration and sulfur nitrogen oxidation, is a bit complicated. So let's see if you've understood which types of electron transfers are going to work for the generation of metabolic energy. Yeah, there's more than one correct answer. So you need to look at your table of redox potentials, or just know which ones are the good electron donors and which are the poor ones, and what is the order. Okay, everybody made up their minds about this? Okay, let's go through them one by one. So glucose as electron donor, nitrate as electron acceptor. Is that going to work? Yes, that's a perfect example of anaerobic respiration. That will work. Glucose as electron donor, carbon dioxide as the electron acceptor. Is that going to work? I would say no. No. Yeah, I think it doesn't happen. So yeah, but yeah, it's a good donor, but with what as the electron acceptor? So I would say this one is not going to work. CO2 as an electron acceptor, it's really the worst. And you can only get a productive metabolism with hydrogen. as electron donor with CO2 as the electron acceptor. Yeah, but that's for the whole thing with the CO2 as the end product. So this is like with the whole reaction of glucose being giving up electrons to release CO2 as a product. Okay, so this is the half reaction of carbohydrate going to the oxidized form of CO2. Now that's very good, but in this case, CO2 is not the electron acceptor. That's the half reaction. And something else is going to have to accept the electrons from this, which is going to be oxygen in respiration, or nitrate, or iron. Okay, so CO2 can never be, never, okay, can only be an electron acceptor from molecular hydrogen. Okay, so this one is no. Nitrite as electron donor, CO2 as electron acceptor. Okay, you say yes. Who else? What else does everyone say? Most people say no. No is correct. Why is it correct? Okay, the half reaction for CO2 is not really up here, apart from like up here somewhere, right? So nitrate is a pretty good electron acceptor. The CO2 is lousy, okay, but terrible. It's the worst. Yeah. So here the question is saying we're going to have a good electron acceptor giving electrons up to a poor electron acceptor. That's not going to work. It's got to be the other way around. So no, this does not work. Can't work. Okay, how about this one? Ammonium as an electron donor, sulfate as an electron acceptor. Anybody figure this one out? Yes, maybe. Okay, how about the last one? Let's do the last one. Molecular hydrogen is electron donor, nitrate is the electron acceptor. Yeah, that's going to work. Very good electron donor, good electron acceptor. It's going to be exergonic, energetically favorable. This one, out of kind of the ammonium nitrate couple and the sulfate hydrogen sulfide couple, which one it get the oxidized form of sulfate and nitrate. Okay, this is the reduced form, ammonium, the reduced form of sulfate, hydrogen sulfide, right? Yeah. So we're saying that in this reaction, the electrons are going to go from ammonium to sulfate. What's the opposite reaction? Sulfide to nitrate. That's if it's going the other way around. One of these is going to work, one of them is not going to work. Which way round does it go? Which one is the better electron acceptor, sulfate or nitrate? Yeah? Nitrate is a good electron acceptor. Sulfate, not so good. So, which way round is it going to go? Yeah. As a donor. Yes, that will work. Ammonium as the donor, sulfate as the acceptor, that will not work. The only reaction that you can use that will be favorable to oxidize nitrogen compounds, ammonium nitrite, has an even better electron acceptor at the end, which is, what did we say? For nitrogen oxidizers, they have to have Yes, have to have oxygen present. Sulfur oxidizers, you have to have a better electron acceptor than sulfate. So it can be nitrate and it can be oxygen. That's what we saw here, right? Sulfide is the electron donor, nitrate is the electron acceptor. That's energetically favorable. Here, we're saying this is going to be the other way around, but it won't work. The delta V is the wrong way around. Okay, so it's not simple, this stuff. I mean, I always got mixed up with it, and actually, when I was learning microbiology, I kind of had to try and learn these things by heart because I did not understand why, in some circumstances, you have nitrification, produce nitrate from ammonium, and sometimes you have denitrification. Nitrate is going to be reduced to molecular nitrogen, N2. And the reason why it works, it depends on whether oxygen is present or not. Okay? This is what makes a difference about whether nitrogen is going to be the electron acceptor or the electron donor. Depends on whether you have oxygen. Okay, so we'll come back to this when we do the nitrogen cycle. Okay, so to put all of this into some kind of context here, I'm going to talk a little bit about the carbon and nitrogen cycles and the role of bacteria in these large biogeochemical cycles, and then we'll finish up with photosynthetic bacteria. So, probably you know all about this, okay? So atmospheric carbon dioxide can be fixed into carbohydrate, and then heterotrophs are going to use up the carbohydrate, oxidize it to carbon dioxide, either by respiration or anaerobic respiration and fermentation. So this side here is in the presence of oxygen, and this is in the absence of oxygen. Now, in terms of carbon fixation, really the really big contributors are oxygenic photosynthetic organisms. So land plants and oceanic microbes. So these will be diatom cyanobacteria. And it's about 50-50 in terms of carbon fixation on land and in the oceans. Now, there is some carbon fixation from autotrophs. Okay, so these sulfur oxidizers, the chemolithoautotrophs, hydrogen oxidizers, sulfur oxidizers, they all fix atmospheric CO2, atmospheric carbon. However, in quantitative terms, this is maybe 9,000 times less carbon is fixed by these pathways. Okay, I don't know why I put boxes around here. Can't remember. Oh yeah, that's right. Yeah, so just thinking about microbes for a bit. Okay, so we all know that all types of animals use organic carbon and perform respiration. So in bacteria, I want to take a little look at what carbon sources can be used by bacteria under what circumstances. And then I'll talk about methanogenesis, which is here, which is an unusual process and is important for worldwide carbon cycles and for global warming. Okay, so microorganisms and carbon use. Okay, so there can be plenty of different carbon sources produced by organisms that fix carbon. So plants can produce starch, cellulose, lignin, and other stuff. So I want to talk about these three that are, okay, that are perhaps quite difficult to digest. So carbohydrates, starch, glucose, everybody can metabolize glucose. Okay? Animals, microorganisms. And microorganisms can metabolize glucose in the presence of oxygen or perform fermentation without oxygen. Okay? Right, cellulose. We cannot digest cellulose. Okay? Try eating grass for a week, you will not like it. Okay? And in fact, no animals on their own can digest cellulose. In ruminants or other herbivores, this enzymatic step, cleaving of the beta 1 to 4 bond between glucose monomers in cellulose, is performed by the symbiotic bacteria. So in ruminants, this is called Ruminococcus species. So Ruminococcus in the guts of cows will ferment cellulose. And their fermentation products will be short chain fatty acids, short chain, yeah, fatty acids like butyrate, lactate, okay, lactic acid, butyrate. These will be absorbed in the gut of the cow as a source of carbon. So that's how, cows get energy from this process. So there's no oxygen inside the cow's guts. So the bacteria have to use fermentation. The fermentation products have still got a lot of chemical energy left in them. So they're absorbed by the cow and used as a source of carbon. That's also true in all herbivores and insects that can also digest cellulose. So lignin, this is a tough one actually. Lignin is what? Yeah, wood. The polymer that makes up wood is lignin. It's very difficult to degrade. And in fact, many bacteria, no, what am I saying? No, bacteria do not digest wood. What kind of organisms break down wood? That's true. Termites can do it. But what kind of microorganisms can degrade wood? If you go in a forest with a tree trunk. Yeah. So fungi, filamentous fungi are the main organisms that digest lignin. And they can only do so in the presence of oxygen. Right? So if wood is buried or somehow stored in an environment without oxygen, then it cannot be degraded. Okay? So this is why, in kind of quite old-fashioned building technique on marshy ground was to use wooden piles driven into the ground, and then you build the building on the top of these wooden piles that are in the marshland. And that is very stable until you drain the marshes. And then oxygen will be present, and so fungi will be able to degrade the wood. And this causes subsidence. So if you look at, can anybody think about some buildings in Nantes where this has happened? Hotel La Pérouse, but that was built especially like that. It's not an old building. It looks like it's on a slant. So, but there's one across from the prefecture that looks just like that, yeah. Another place, the one I'm thinking of, that used to be next to water, but now there's no more water there. Well, hey, come on, hands up, who's from Nantes? Come on, admit it. Who is from this town? Yeah, some people. A place in the center of town with some old buildings, There used to be water around there, but there's no water anymore. Used to be an island. It's still called the something island. L'île Fédot. Okay, so if you take a walk around the Île Fédot, you'll see that some of the buildings, they're kind of like leaning over like that. That's because they were built by, anybody know their non-local history? Who built these buildings? The slave traders, but it's near to the Place de la Petite. Yeah, Dutch traders. So they built all these buildings just like in Amsterdam with the wooden pylons under the pile underneath to support the building. So this is all built like this. It was on an island, so where the wooden piles were was under the water table. There was no oxygen. When this all got drained to fill in the part of the river, then you've got oxygen attacking the foundations, and that's why the building subsided. Okay. So that's the lignin story. And the other thing that can be quite useful for bacteria and carbon use is synthetic hydrocarbons that are produced by the chemicals industry. Could be just toxic aromatics, or what is another big problem worldwide at the moment? Plastics. Okay, so some simple hydrocarbons can be degraded by bacteria with oxygen or by anaerobic respiration with nitrate as electron acceptors, but plastics can generally not be degraded. However, if you look carefully around sites that are polluted, then there are some bacteria that have been found that can degrade polyethylene, PET, this kind of stuff. So there's perhaps in the future some way to get rid of plastic pollution by investigating these bacteria, understanding the enzymes that degrade plastics, and trying to get rid of them that way. One of the drawbacks, if we were to do this, might be that these bacteria would release the carbon dioxide. Because at the moment, in the plastics, this carbon is kind of fixed. And so you have to be careful whether we really want to oxidize all the plastic that is around. Probably in the oceans, yes. If we could get rid of it, that would be a net benefit. Okay, so I'm just going to talk a little bit about methanogens, also in relation to global warming, because this could be a significant contributor to greenhouse gases. So methanogens. So the basic reaction is using molecular hydrogen as the electron donor, carbon dioxide as the electron acceptor. And this will work, okay? Because your delta V is going to be positive. And the only bacteria that can perform this reaction are archaea, Methanococcus methanosarcina. Okay, the delta V is positive, but it's really pathetic, okay? Not even 0.2 volts. So it's really tough to try and squeeze out some metabolic energy from this reaction, enough to make ATP or anything else. Only methanogens can do this by kind of like complex chemistry. And it occurs in anoxic environments where you have a lot of organic carbon. For example, in ruminant guts. The cow puts in the carbon, and the gut is maintained without oxygen. Marshes, okay, you get methane produced as marsh gas. And another big site of production is are the sediments on the continental shelf. So if you've got coastal biosystems which have a lot of animals, plant life living there, as they die, these organisms die, they'll float down to the bottom and start to sediment out underwater. And methanogens live in this environment and they release methane. But it's down in the bottom of the ocean, it's cold and it's under high pressure. So this forms methane clathrates. So it's like, or methane hydrate. So it's frozen methane and water complexes. Okay, so this methane is not released into the atmosphere. Yeah, it's stocked. But one of the worries is, there's also apparently some in kind of like permafrost environments. So one of the worries is if you know the ocean starts to warm up by a couple of degrees, these methane hydrates will release methane gas, and there'll be a kind of spike of methane released into the atmosphere. This would be very, very problematic because it's a 25 to 30 times more powerful greenhouse gas than carbon dioxide. So if you have a sudden release of methane into the atmosphere, then this is going to increase our problems. But it would be a short-term effect because methane is not stable in the atmosphere. It will be oxidized to carbon dioxide. So the half-life is much shorter than CO2. Half-life of CO2 in the atmosphere is like massive. You know, it's thousands of years. Methane, I don't know, 10, few decades. Yeah? Okay. Actually, I am not a big expert about this, so maybe you should take my numbers here. Maybe my numbers are not good for the half-life of CO2 in the atmosphere. So if you have other information, that could be right. Methane has a shorter half-life than CO2 by a long way. So that's definitely true. So is this methane, is it all going to be released? Well, perhaps not, because as well as methanogens, there also exist bacteria which can consume methane, which are methanotrophs. And methanotrophs can also exist in an anaerobic environment because they live as consortia between the methanotrophic bacterium, which is an archaea, and sulfur-reducing bacteria. So overall, you've got methane as the electron donor, sulfate as the electron acceptor. Think about this. This will work in energy terms, okay? Because, yeah, sulfate is a better electron acceptor than carbon dioxide, so it should work. But in fact, each of these bacteria has got half of the enzymes that are required to make this work. So each one of these species on its own cannot perform this metabolic step. So they only work as consortia. So they cooperate between species. So, and this involves electron transport from the methanotroph. Okay, so the electrons come from here, they're transferred from this bacterium to the other one, and then onto sulfate. And it was recently found how this occurs. So this is a Nature paper from 2 years ago. And the authors cultured, this is the archaea, here is with A, and the sulfur-reducing bacterium is H. When they were cultured together in the presence of, I think they used, they found some way to get methane generated as a carbon source. What happened is that the sulfur-reducing bacteria, which were gram-negative, started to produce type 4 pili that made connections with the archaea. And these pili functioned as nanowires. So the electron transfer, kind of schematized here, was actually through these little tiny wires, pushed out by the sulfur-reducing bacteria. Okay, so that's how methanotrophism works. So maybe these methanotrophic consortia will save us from a sudden release of methane because they live in the same environments. Okay, they live off the methane that's released by the methanogens. Slightly higher up in the under sub-ocean sediments, where you still have some sulfate, but you don't have any oxygen. Okay, and methanogens are going to be even deeper when there's no more sulfur around, no more sulfate around, and there's only carbon dioxide to be used as an electron acceptor. Okay, so maybe methanotrophs are going to save us all. Okay, the nitrogen cycle. This is this horrible, complicated diagram that I copied from Prescott's Microbiology, the textbook. So what have we got? Okay, oxidized nitrogen, nitrate, reduced nitrogen, ammonium, and then somewhere in the mix, nitrogen gas. Okay, so we're going to have organic nitrogen in amino acids, for example, purine pyrimidine bases, but let's just say amino acids. So all organisms, all bacteria, all the rest of us, we want to have this. Okay, we've got to get this. So if ammonium is present, okay, that can be assimilated easily into organic nitrogen. If nitrate is present, many bacteria can perform assimilatory nitrate reduction and get organic nitrogen. On the other hand, we have these other arrows, the purple arrows and the white arrows. Now, out of these processes, which of them are being used by bacteria to generate metabolic energy without organic carbon? Which arrows represent that? These guys. Hang on, I said no organic carbon, but let's say this in the presence of oxygen. Which arrows represent that? The white arrows, okay? So you've got ammonium oxidized to nitrite, that's nitrous amonas, nitrite to nitrate, nitrobacter. So this is ammonium oxidizers, chemolithoautotropes. Will work only in the presence of oxygen. And the bacteria are doing this to generate metabolic energy. Okay, that's these white arrows. So the purple arrows, what is happening here? The opposite, right? So what is happening here? Where's the energy source and what is the role of the nitrogen in this process? Electron acceptor. Here, the ammonium is the electron donor, oxygen is the electron acceptor. If things are going the other way around, then the nitrate is the electron acceptor. What's the electron donor? It's not on the diagram. But what the bacteria, okay, no oxygen, you use nitrate as the electron acceptor. What process is this? Can we just, yes, anaerobic respiration. So you have organic carbon as the energy source. You produce, it's getting oxidized. You produce NADH, and the electrons go onto nitrate. Okay. So this is what's going on with the purple arrows. Okay. So the oxidized forms of nitrogen are used as electron acceptors in this case for anaerobic respiration. So some bacteria can reduce them all the way around to ammonium. in terms of having ammonium available to produce amino acids, that's okay. But denitrification is what we saw with the Paracoccus denitrificans. So the anaerobic respiration will produce nitrogen gas as a product. And that leaves the rest of these arrows here and these ones, which are kind of weird. This one, nitrogen fixation, the assimilation of atmospheric nitrogen gas into organic nitrogen. Who performs this? Okay, here we go. We've got the names of these guys down here. Photosynthetic bacteria, cyanobacteria, perform nitrogen fixation. Not on the list here are the ones that are the most important for us, which can be what? Where do you find nitrogen fixation and is very important for human beings? Nowhere. Okay, if you go back to the Middle Ages, medieval period, people would rotate crops on their land to maintain fertility. Yeah, fertilizer. But what kind of plant produces natural nitrogen fertilizer? If you want to keep your land fertile, you rotate the crops and plant some. Nobody knows. Okay. Beans, peas, legumes, right? These are nitrogen fixers. But it's not the plant that does this. They can only fix nitrogen because of their association with symbiotic bacteria in root nodules, which are genus Rhizobium. Okay, so these bacteria fix nitrogen. They associate only with the roots of leguminous plants. So other plants also have symbiotic associations with other nitrogen fixers. Some trees associate with Frankia genus of bacteria, which also fix nitrogen. So the first trees that can grow back on a very poor soil, they'll be like Les Aulnes, older trees. And these fix nitrogen through their symbiotic bacteria. Now, this nitrogen fixation reaction is it's just amazing that it can ever happen because it is so energetically unfavorable. Nitrogen gas in the atmosphere is fantastically stable. You know, you can't like set it alight and burn it or something. It just doesn't work. It's incredibly stable. Okay, let's see if you know anything about chemistry, you guys. What process is used industrially to produce ammonium from atmospheric nitrogen? Anybody know? Nobody knows. Huh? One. Yes. The same guy, but same inventor, but different reason for inventing the process. Okay. First, let's talk about the process first, and then I'll give you my version of First World War history, okay? So the chemist did this. What? Yeah, the Haber process. Right. So the Haber process, it allows the production of ammonium from atmospheric nitrogen gas. It involves heating the whole thing up, heating the two products up at like 400 to 500 degrees at high pressure with a metal catalyst, osmium or iron. So in order to get this reaction to go chemically, you need a huge input of energy and a catalyst. Otherwise, it just won't work. So it was just amazing that anyone could get this to work synthetically. So this is a very, very, very difficult reaction to perform. So it's quite incredible that any enzymes can do this. Okay, but there are so nitrogenase is the enzyme that does this, and even though different, very different types of bacteria perform the nitrogen fixation, the nitrogenase enzyme is highly homologous in all the different genera that perform nitrogen fixation. So it's probably been transferred from one bacterial species to another. Okay, so it's one appearance of nitrogenase somewhere and then transferred to other bacteria. It's very sensitive to molecular oxygen because the process involves transferring electrons onto dinitrogen gas, and the iron-sulfur clusters that perform this electron transfer can easily be oxidized. So this reaction can only work in the absence of oxygen. Okay? So during the production of root nodules in leguminous plants, it's the plant which will provide the anaerobic environment for the bacteria that allows nitrogen fixation to occur. And this works because plants produce leg hemoglobin in their root nodules, which fix all the oxygen that's around. Okay, they suck up the leg hemoglobin, binds all the oxygen. Okay, so that's nitrogen fixation. Rhizobium, cyanobacteria are the major types of bacteria which perform this, and of course very important for us for agriculture. It's thinking about this. Yeah, just one more. Oh yeah, so I was going to go back to World War I, right? Okay, so why was the Haber process or the Haber process very important for Germany? It was important because okay, who had the biggest navy 100 years ago in the world? The British, yeah. British Navy. We had a massive empire, huge, very important maritime power. So one of the first things that happened at the outbreak of World War I was the naval blockade of Germany by the British fleet. And this was very serious for Germany because they had to import nitrogen fertilizer. And this meant that agricultural production in Germany was going to go down. And it's really Fritz Haber that allowed Germany to hold on for 4 years, because the Haber process allowed the production of ammonium, which could be used to produce as fertilizer. So I don't think the real objective was to produce poison gas. He did do that, actually. He produced chlorine, and he was responsible for the industrial production of chlorine to be used as the first gas in World War I. Okay, to my mind, I think it's 2 different projects, because you can't produce chlorine gas if you're trying to produce ammonium out of atmospheric nitrogen, right? So, and then, okay, other second-generation poison gases, nitrogen mustard, might have been produced afterwards because you had the ammonium to do it. Okay, another little digression here is Haber's wife killed herself because I think Clara, she was called. She shot herself because she thought it was wrong to use scientific advances to kill people in this way. Okay, what else to say? Yeah, so that's nitrogen fixation. And the last section here is, yeah, I forgot about all of this. So the last section is these arrows here, which are called the Anamox process, which was discovered in 1999. Okay, so Anamox is for anaerobic ammonium oxidation. And these bacteria are planktomycetes, so weird bacteria, chemolithoautotrophs. And what they're doing is, in a weird way, using ammonium as the electron donor, nitrite as the electron acceptor, and this will produce dinitrogen gas as the waste product. Okay? And they grow very slowly. It's an inefficient process because, okay, the energy, the thermodynamics are not great. But it has a lot of potential. So this apparently is responsible for maybe 50% of the denitrification that occurs in the ocean because of these anammox bacteria. So previously it was thought that denitrification by anaerobic respiration was what was removing nitrate and ammonium from the nitrogen cycle. But in fact, the anammox process is important environmentally. And it's also important for industrial applications, for water treatment, okay, because you can't throw out wastewater with a lot of ammonium present. Okay, so sewage water, it's got a lot of organic nitrogen. If at the first stage you'll have aerobic bacteria, or you have anaerobes using the organic nitrogen, it will be transformed to ammonium. And then in the next stage, you have these aerobic ponds, so they're stirred, you have air bubbling through them so that you can have nitrification. And then afterwards, you have another anaerobic step with bacteria that will use the organic carbon and denitrify it to release nitrogen gas. So this process of water treatment to get rid of the ammonium and the nitrate is very effective, but you need to have oxygenation and carbon input. Whereas if you have the anammox process working, you don't need to add any organic carbon because they're chemo-litho-autotrophs, and you don't need to have oxygenation. So you don't need to stir the whole stuff and have air bubbling through it or anything. So it could technically be much more effective as a way of getting rid of nitrogen from wastewater. Not actually widely used yet because it was discovered too recently. So there's pilot systems in Holland. Okay, so we have about, let's say, 15 minutes left, maybe 20 minutes to think about bacterial photosynthesis. Phototrophs. Okay, so what is the message here from this slide? There are many different types of photosynthetic bacteria. Okay. Among the eubacteria, they all use bacterial chlorophyll as the light harvesting pigment, whereas photosynthetic archaea use bacterial rhodopsin. So because you've got two different chemical systems, then bacterial rhodopsin and bacterial chlorophyll seem to have evolved independently. However, bacterial chlorophyll seems to have evolved once, but look at this. You don't have any phylogenetic clustering of these photosynthetic bacteria. So how did these bacteria become photosynthetic? Well, probably because of lateral gene transfer. Somewhere, one of these groups of bacteria developed bacterial chlorophyll and the photosynthetic system, and it got transferred to the other groups. Okay? So that's why you have phylogenetically diverse bacteria which are photosynthetic. Now, out of all these different systems, only cyanobacteria perform oxygenic photosynthesis, the one that we all know in green plants. So all the other photosynthetic bacteria do something else. So that's what we're going to look at. And the objective for you is to try and understand what photosynthesis or how photosynthesis contributes to the energy metabolism of these different bacteria. Okay, so Halobacterium salinarum, bacteria rhodopsin. What happens here? It's a 7 transmembrane helix protein. it's in the plasma membrane. So this is the cytosolic side, this is the periplasmic side. When light hits the photoreactive center here, bacteriorhodopsin, then this will cause the whole protein to pump a proton across the membrane. So it's a kind of light-activated proton pump, so you get proton motive force. No electron donor, no electron acceptor. The protons will go back across the membrane via the proton-dependent ATPase. So photosynthesis allows the bacterium to generate ATP without any kind of input of carbon or anything. Okay, that's what I was saying before. Multiple gene transfer between phototrophs. It's kind of difficult to really find out what was the first photosynthetic bacterium, but just don't really need to remember this. So you have these different groups, Chloroflexi, Chlorobi, Helibacillus, Propylbacteria, which are getting different bits of the photosynthetic apparatus, either photosystem 1 or photosystem 2. And only cyanobacteria have both of them. Okay. So I'm going to give you one example of photosynthetic bacteria with photosystem 1, one example of bacteria with photosystem 2, and then cyanobacteria. So Chlorobi, they are the green sulfur bacteria. Green non-sulfur photosynthetic bacteria are Chloroflexus. What is happening in Chlorobi? Okay. Photosystem 1. Okay. Right. So the requires an electron donor, which is going to be hydrogen sulfide. This electron is transferred onto the cytochromes and fed into the reactive center of photosystem 1. When light hits this, that electron is going to be transferred, it's going to gain energy. Okay, so it's transferred onto another molecule, and then it can be transferred down onto NAD+ to generate NADH. What does this mean? This means that because you add energy in, you can transfer the electron from sulfide to NAD+. Normally, this would not happen. Okay, because think about the chemolithotrophs. Sulfur oxidizing bacteria, they can't transfer the electron from hydrogen sulfide directly onto NADH. If you need to put energy in to make that occur. So that's what the utility is for these bacteria. It makes the energy metabolism more efficient. So one of the questions about photosynthesis and its evolution is, yeah, well, the whole thing is so complicated. How on earth can, if it's so good, how could any kind of bacterium exist without photosynthesis? Because if you think that photosynthesis has evolved from somewhere, then there must have been something living without photosynthesis before. Well, here's the answer. Chlorobi, before they became photosynthetic, they were sulfur oxidizing bacteria. They were chemolithoautotrophs. They gain photosystem 1, and they can become photolithoautotrophs. Okay, purple non-sulfur bacteria. So these guys. They only have a type 2 photosystem. What happens in a type 2 photosystem? Well, light hits the photoreactive center. The electrons are, energized, and then they are transferred onto Q. It's this guy again. What is that? Quinone. Quinone, coenzyme Q. So you have the quinone cycle. Quinone cycle generates proton motive force because it picks up protons on the cytoplasmic face of the membrane and releases them on the periplasmic face. So here, you've got no electron donor, no electron acceptor. But the photosynthetic system is allowing the bacterium to generate proton gradient. So it's a little bit like what is happening here, okay? No electron donor, no electron acceptor, but there's a kind of like a cycle that allows you to generate a proton gradient. So that's what photosynthesis is adding to the bacterium here. It can, generate ATP from nothing, just from light. And cyanobacteria, this is the combination of both photosystem 1 and photosystem 2. Okay. I don't want to go into this because, it's the same thing as in green plants, and you'll be doing this in plant physiology, I guess. So for phototrophs, different types of photosynthetic bacteria. Light is the source of metabolic energy, but what is the source of electrons? What is the source of carbon? Okay. So for chlorobene, they are photolithic autotrophs, but anaerobic. Source of electrons, reduced sulfur or molecular hydrogen, carbon source, carbon dioxide. Chloroflexus, green non-sulfur bacteria, they are photoheterotrophs. They use organic carbon. Purple sulfur bacteria, okay, is a bit like chlorobe, anaerobic photolith autotrophs. Purple non-sulfur bacteria, so these are, this is chromatium, I think, and these are, rhodospirillum, or rhodobacter. Is that what I said? Yeah, rhodobacter, purple non-sulfur bacteria, rhodobacter. These are photoheterotrophs. And in fact, if you culture rhodobacter in the dark with organic carbon, they grow quite happily. They lose their color, they grow as chemoheterotrophs. So in this case, you've got photosynthesis, which has been grafted onto a kind of anaerobic chemoheterotrophic type of lifestyle. And cyanobacteria, okay, they're both photosystems. Water is the electron donor, and they are chemolithoautotrophs. Okay, so here is a kind of summary of everything. Respiration. Everything starts with organic carbon. Then electrons are transferred onto an ADH. Electron transport chain gives you proton motive force and gives you ATP. And you really should understand the difference between anaerobic respiration and fermentation. Chemolithotrophs, how are they working? Well, the basis of their metabolism is some kind of electron transport from an inorganic donor to an inorganic acceptor. Okay, yeah, that's true. So you start out generating energy by an electron transport mechanism. This will allow you to get proton motive force and ATP. It can also allow you for nitrogen oxidizers to produce NADH by reverse electron transport, and then you use that to fix carbon and generate organic carbon. And the big problem of this is to understand why only certain couples of electron donors and acceptors will work in energetic terms. And for phototrophs, try and understand, what these photosynthetic bacteria were before they gained photosynthesis by lateral gene transfer. Okay, so I guess that is about enough bacterial energy metabolism for any rational person. Just before I leave you and let you go away, I'll give you the scores for Microbe of the Week so far. The participation has been, let's say, patchy up to now. So, okay, here are the scores, okay. Only a couple of people answering both questions so far. And, okay, so there are the scores. We have like Leah and Rafik there making the running here. And a few people in the pack behind and a whole bunch of people just not interested. Okay, so I try and give feedback if I can. So for next one, when would you like me to send a question? When would be a convenient time for participation for you guys? Sunday morning? What do you think? Sunday morning. Well, everybody okay with for Sunday morning? There's no time limit, but it just says the bonus points for the first correct answer, right? Yeah, until the next lecture. Well, which is going to be ages away, but yeah, let's say a week. You can still try and answer the previous ones if you like. I'll count the points. So there's no time limit, but it's just if you want to win, you should try and answer quickly and get the first correct answer. Okay, yeah, so Sunday morning, is everybody okay? No, is it a joke? This evening? No? No? Yeah, so... Huh? Sunday evening. Okay. Okay, let's have a vote here. We've got 3 options, right? Okay, 3 options. This evening. Who says yes for this evening? Yeah, he's a delegate, so he has all the power, right? Exactly. Sunday morning. Who says okay for Sunday morning? 3, 4, 5 people. Sunday evening. Who prefers Sunday evening? Oh man, it's a split decision. So, actually, I prefer Sunday evening because often I check work for the next week at the end of the day. So, Sunday, what time? 6 p.m.? 7. Sunday, 7 p.m., okay? Or Sunday, 7 p.m. for microbe of the week.